Hi everybody, my name is Govindaraj Dev Kumar, or Govin for short, and I am an assistant professor at a Center for Food Safety at the University of Georgia's Griffin campus. Right now I'm in India and I'm on a mission to look for this microorganism called Salmonella typhi that causes typhoid fever from food and environmental samples in India. So if you hear birds chirping and noises of cars honking, my apologies in advance. So this seminar is about antimicrobial sanitizers and disinfectants and their uses for improving the safety of foods. So as I said previously, I am an assistant professor at the Center for Food Safety, which is at the University of Georgia's Griffin campus. I am a food safety microbiologist, and one of my main areas of focus is adaptation of foodborne bacterial pathogens to sanitizer stress. In particular, I look at a phenomenon called filamentation, which is where bacteria, they it kind of like a centipede. Bacteria start forming these long chains of undivided cells that once the sanitizer stress is removed, kind of increase in number when the cells separate from that centipede or the chain link. So that's it. And this phenomenon is seen in a lot of different types of foodborne bacterial pathogens such as Salmonella, Escherichia coli, and it's fascinating because it's a transient phenomenon. So it's formed for like an hour or two, and then it disintegrates with a massive spike in population. Uh, so where I apply this is produce safety. So Georgia, like Texas, is a state that does a lot of agriculture, and Georgia is known as a peach state. So currently, I'm working on understanding how sanitation practices help in reducing microorganisms in peach processing facilities in Georgia. So for this talk, we will look at the definitions of basically what is a sanitizer, how is it different from a disinfectant, or something, how is it different from something like gamma radiation, which is a sterilant. We'll look at some of the commonly used sanitizers in food safety, such as chlorine or peroxyacetic acid, and we will understand what is their differences in the mode of action. Why do we use this? One of the other focuses of this will be the history of sanitizer use, which is fascinating because before sanitizers came into prominence, diseases were rampant, and they really have a gnarly but an interesting history. We'll look at some of these tests to determine sanitizer efficacy. So how do we find out which sanitizer is the best and how strong is the sanitizer? We'll also look at how microbes become tolerant or resistant to sanitizers, and then some of the regulations that the EPA or the FDA have associated with sanitizer use, and then briefly touch upon sanitizers that can potentially become commonplace in the future and replace currently used sanitizers. So here we will discuss the broad array of categories that you have for these chemical agents that we use for cleaning, sanitizing, and disinfecting surfaces, and even sterilizing surfaces. So when we go shopping for uh, you know for something to clean our kitchen or something, we might come across detergents. Detergents can be applied to surfaces to remove the soil, so to remove like dirt, grime, debris where bacteria might be attached. And cleaners are sometimes used with physical forces, such as, say, washing or brushing. Then we would, once you clean a surface, you would use a sanitizer. A sanitizer reduces the number of bacteria. So, examples of sanitizer would be like chlorine. A disinfectant is a little stronger than a sanitizer. It specifically kills a higher number of fungi, bacteria, or viruses. And what is important here is the word kills. A viricide or is more particular in its mode of action. Disinfection you can reduce fungi, bacteria, or viruses, whereas in a viricide, by as its name suggests, is important for reducing the number of viruses. Again, like a sanitizer, you can have like a 3-log reduction. A viricide can also give you different levels of reduction. And then a sterilant makes a surface sterile, like eliminates all microorganisms from growing back. And not only are these for vegetative organisms, like say like bacteria, viruses, but also their spores. So fungal spores, bacterial spores are also destroyed when you use the sterilant. An example of a sterilant would be something like ethylene oxide. So now let's look at some definitions that will help us categorize these chemicals better. So what is sanitize? The sanitize means to reduce microorganisms of public health importance. Now, what are microorganisms of public health importance? In a food safety context, it could be something like salmonella or Escherichia coli or E. coli. It could also be something that can cause spoilage of food and then the person becoming sick because of the spoilage of food. So that could be something like Pseudomonas. And it says important to levels considered safe. So what does levels considered safe mean? Well, you need a certain amount of microorganisms to cause a disease. Say if you need 1,000 salmonella cells to cause a disease, and you have 1,000,000 salmonella cells on a chicken carcass or poultry carcass, you want to reduce it so that it's less than 1,000. So if the consumer consumes it, they do not fall sick. Um, so this is under established parameters. So like, should it be a surface with soil? Should it be stainless steel? All of those, or should it be meat? Then without adversely affecting either the quality of the product or its safety. So you don't want to, if it's a surface, you don't want to destroy the surface. You don't want to cause corrosion or pitting. If it's a food surface, you don't want off flavors or oxidization or oxidation, and you don't want browning or damage, tissue damage. So what is a sanitizer? A sanitizer is a substance or mixture of substances. So substance could be something like chlorine. A mixture of substances could be peroxyacetic acid, where you use the organic acid and an oxidizer. Then this reduces the bacterial population in the inanimate environment by significant numbers. So 3 log 10 reduction. Now what is a log 10 reduction? 1 log is 90%, 2 logs would be 99%, 3 logs would be 99.9%. So if you have 1,000,000 cells, you can imagine, well, not imagine, but you can actually calculate how much a 3 log 10 reduction would reduce from those 1,000,000 cells. But a sanitizer does not destroy or eliminate all bacteria. Like we said, it does not completely remove all the bacterial cells. And remember that we are mentioning bacteria here, so it's specific. It does not kill viruses or spores. Sanitizers, meaning public health ordinances, are used on food contact surfaces and are termed sanitizing rinses. So these are very important to the food industry because they prevent bacterial buildup and they prevent bacterial foodborne pathogens from actually surviving on these surfaces. So the EPA defines sanitizers as chemicals that kill bacteria on surfaces and are not intended to kill viruses. An example of a sanitizer would be chlorine. Now we come to the next batch of definitions we now move on to something that's stronger than a sanitizer, and this is called a disinfectant. So what is the meaning of disinfection? To disinfect means to destroy or irreversibly inactivate specified infectious fungi and bacteria, but not necessarily the spores on hard surfaces. Now, compared to a sanitizer, we can see that a disinfectant seems like it is stronger, it is more broad spectrum, but it does not seem to kill spores. So, what is the definition? A disinfectant is a substance or a mixture of substances, exactly like how we had defined a sanitizer, like chlorine and peroxyacetic acid. Disinfectant could have a mixture of substances, and these need to destroy or irreversibly inactivate bacteria, fungi, and viruses, but not necessarily bacterial spores in the inanimate environment. So EPA registers three types of disinfectants based on the type of efficacy data submitted. So limited, general, or and hospital. So limited would be for a particular organism. So you might have a disinfectant that works particularly against salmonella, and so it can be used in processes or environments where salmonella is a major problem. A general disinfectant will work against both gram-positive and gram-negative bacteria. An example of a gram-positive microorganism or bacteria would be Staphylococcus aureus. An example of a gram-negative microorganism would be Salmonella enterica. And then hospital. Hospital infections are called nosocomial infections, and we will discuss in the history of sanitizers of why um, our efforts to control infections in the hospital resulted in the development of basically sanitizers and all these protocols to reduce infection control. An example of a disinfectant would be alcohol. And continuing with definitions, now we'll see what an antimicrobial is. An antimicrobial is a very generic, broad spectrum term. So antimicrobial drugs include all drugs that work against a variety of microorganisms, such as bacteria, viruses, fungi, and parasites. So if you have an infection or you want to reduce a microbial population, you would use an antimicrobial. When you use an antibiotic, an antibiotic is more specific. It is a drug that is effective against bacteria. So all antibiotics are antimicrobials, but not all antimicrobials are antibiotics. This means that antimicrobials are more broad spectrum and they're not only associated with bacteria. Now, antiseptics are a substance that is used to destroy pathogenic organisms. So, an example would be iodine for to clean up wounds, or povidone iodine, and then a sterilant. A sterilant kills all microorganisms from a treated surface. Sterilants are very important for like surgical instruments, and they're like even masks. Sometimes things like lab coats are put under sterilants or sterilizing treatments to reduce the risk of someone carrying these pathogens from one place to the other. Now, these are the broad definitions of antimicrobial sanitizers that one would come across in the food industry. In our next set of slides, in the next lecture, we will look at how the history of antimicrobial usage helped in us understanding infections and helped in the development of infection control protocols. Thank you.